Hi everyone, and thank you for joining today for another edition of our bite-sized learning series where we will discuss genome-wide CRISPR knockout screens. My name is Lauren Fait, and I'm a senior sequencing specialist here at Illumina. Today, we will walk through an overview of what a genome-wide CRISPR knockout screen entails, as well as a few factors in order to ensure success. So let's go ahead and get started. The ability to modify the expression of single genes has become one of the most important tools in molecular and cellular biology. And so several methods, such as CRISPR, have actually been developed to allow for specific gene manipulations in cells. Some common applications of CRISPR technologies can actually be seen in the figure to the right of this slide here. However, in this presentation, we will focus on what is called CRISPR screening, which is essentially a large-scale genetic loss-of-function approach designed to find the equivalent of a few needles in a haystack in the world of biology. And we will mostly focus on pooled CRISPR knockout screens, but this method can also be employed with transcriptional inhibition or activation with CRISPR-I and CRISPR-A. Now, CRISPR screens allow you to discover key genes that elicit a specific function or phenotype under investigation for a given cell type, while starting with a very broad hypothesis, or in some cases, almost all of the genes in the genome. And so these CRISPR screens then enable you to generate a new narrow hypothesis, as what you're able to get from a genome-wide CRISPR screen is a short list of candidate genes or sequences that produce that physiological response under investigation that can then be investigated further. And so, for example, these screens have uncovered genes responsible for survival, proliferation, drug resistance, viral susceptibility, and additionally, some other applications that you can see listed to the left of this slide here. And this technology has also even been applied to the functional interrogation of the non-coding genome as well. And so really what you can see here is that the possibilities are endless for these CRISPR screens. But before we dive deeper into genome-wide CRISPR knockout screens, let's quickly review what CRISPR is and the components of CRISPR as well. And so CRISPR was initially discovered as a prokaryotic adaptive immune system. However, now this system's been adapted into mammalian cells to generate a simple and efficient means of creating targeted loss of function mutations, among many other applications that we will not discuss in this presentation. Cas9 proteins are able to generate precise double-stranded breaks at target loci determined by short guide RNAs or sgRNAs. And so in the absence of a homologous template, these breaks are repaired by an error-prone non-homologous end joining mechanism, which can then lead to the production of indels, which then cause frame shift mutations, leading to the knocking out of our genes of interest. Now, there are a few things needed here in order to generate these knockouts. And so the first thing needed here is your nuclease or your Cas9. And so this must be expressed and translocated in the nucleus of your cell in order to function. And then the second thing you need here is your sgRNA or your guide RNA, which is going to direct Cas9 to a specific site in the genome to cut. And so guide RNAs are typically comprised of a cRNA sequence that is a 20 nucleotide target specific sequence that's going to recognize that target in our genome of interest, and a Cas9 binding handle sequence, or where our nuclease Cas9 is going to bind. Now, a prerequisite for the Cas9 to cleave the target site is the presence of what we call a PAM motif, and this is going to follow that target specific sequence. And so the PAM sequence is actually required for a Cas nuclease to cut and varies with the endonuclease being used as well. So that's something you're going to want to keep in mind when designing your guide RNAs or your CRISPR experiments. Now, the cool thing here with CRISPR when compared to other gene editing technologies like zinc finger nucleases and transcription activator like effector nucleases is that you don't need to engineer a completely new DNA binding protein for each target. Instead, because the system utilizes an RNA-guided nuclease, all you need to do is synthesize or design a new piece of RNA complementary to your target, which dramatically simplifies and greatly reduces the time needed for gene editing design and implementation. And so because of this, CRISPR-Cas9 has become a really powerful tool in these large-scale loss-of-function screens where we can effectively target almost all of the genes in the genome seamlessly. Now, before we get into the specifics, let's walk through the entire CRISPR screening workflow here. And so the basic idea here is that we're wanting to first produce lentiviruses that are going to contain all of our unique guides, and then we'll use those viruses to deliver the CRISPR cargo to our cells of interest. And so following that viral production, we will then transduce our cell line of interest in bulk with these viruses we just made. And then we'll apply a selection pressure and finally sequence the leftover cell's guide RNA via Illumina sequencing to perform what we call hit identification. And so in other words, we are just PCR amplifying our guide sequences contained in each cell and quantifying which guides are left over in our sequencing data after that selection, and then we rank them. And then lastly, what we can do from here is perform further validation of our potential ranked candidate genes by a small-scale repeat of the experiment to confirm that the genes identified produce the phenotype under investigation, B, sequence-based verification that the target gene or genes were altered in the screen to begin with. So again, we could sequence here via Illumina sequencing. And then C, or lastly, by restoring the wild type gene sequence to see if this rescues the selection phenotype, for example. And we will not get into too much detail here on the validation side in this presentation, but at this stage, you might also want to look at assessing potential off-targets further as well. Now let's focus on section A from the last slide first here before we get into the screen itself, which is actually creating those lentiviruses that we will use for our screen. And so the first thing you're going to need to do is to design your guide RNA library, and you can either do this custom or purchase a pre-designed library from somewhere like Addgene. Now, if you are going to be designing from scratch, you are going to want to make sure that you have assessed any potential off-target effects of your guides using methods such as in silico tools to ensure that what you're observing is not going to be confounded by these off-targets. Another thing that you're going to want to keep in mind is that you'll want to have several guides targeting the same gene so that you can help minimize some of those confounding effects of off-targets to help confirm true positives from false positives, and B, to also reduce the impact of potential guide RNA dropouts. Now, after you have designed your guide sequences, you will then order these as oligos from somewhere like Twist or IDT, or even make them yourself with technologies from companies like DNAscript. Now, the next step here is to get these oligos cloned into plasmids that are compatible with viral production. However, if you purchase a pre-made or designed library from somewhere like Addgene, you'll actually be able to skip this step usually because usually the library is supplied as a pool of plasmids already. Ultimately, though, you are going to want to get those plasmids into bacterial cells through something like electroporation, and you will allow these transformed bacteria to grow and replicate in order to amplify up the library. And now the last step here is to create our viruses, which will be used to transduce our target cells of interest in our screen, and each of these viruses will each contain their own unique guide RNA from the initial pool. And so in order to do this, the guide library will be transfected into viral producing cells, and we will then collect those assembled viruses in the supernatant. And so now at this point, we're actually ready to begin our functional CRISPR screen. Now, we just talked about needing to amplify up our plasmids in those bacterial cells on the last slide. But one of the most critical QC steps here to include in your screen is to ensure that your library amplification was successful and that all plasmids were amplified without any being lost as the bacteria transformed with the library grow and replicate. Additionally, we want to make sure that we are starting our screen with an even representation of all of our guides as well, and that we don't have over or under representation of any of them. And so the best way to do this is to include a sequencing step for quantification of the plasmids to ensure that all of your plasmids have amplified evenly and to ensure that no plasmids were lost or amplified preferentially, or that you are not missing or overrepresenting any one guide, for example. And so to do this, you're actually going to extract the DNA from your colonies and sequence your plasmids via methods such as Illumina sequencing to identify your target guide RNAs. And so typically, it's also helpful to sequence both pre and post amplification so that you can compare the numbers or populations of guides before and after as well. And so you're going to then rank the plasmids by abundance scaled to 1 to create what's called a cumulative wealth distribution curve or a Lorenz curve. And then by calculating the area under the curve, you can get a sense of the uniformity of library distribution, with an AUC of 0.5 being equivalent to perfect uniformity, which is shown in figure A here. But ideally, what you want is an equal AUC before and after amplification, which is shown in figures B and C here. All right, so now that we have a better understanding of how we make these viruses to be used in our screens, let's chat more about the screen itself now. And so the idea here of these genome-wide CRISPR screens is to knock out every gene that could be important, but we only want to knock out one gene or target per cell. And so in other words, we only want one guide RNA per cell. And so really here, the intended result is creating a population of cells with a different gene knocked out in each one of them, again based on that unique guide RNA. And as a result, some cells will die, but others will actually survive or even grow better than before. And so what we do then is follow this up with Illumina sequencing to identify what particular guides may be present or absent from our population of cells. And so we are basically going to be counting these guides in the end here to help identify previously unknown genes that contribute to a particular phenotype. And so, for example, identifying genes conferring sensitivity to a particular drug or treatment. Now, a critical component of a CRISPR screen is the transduction step, and it is crucially important to ensure that you only have one virus per cell, and therefore only one guide or knockout per cell. And so in order to do this, you're going to use what we call a low multiplicity of infection, or in other words, you're going to use an excess of cells that will not actually end up getting transduced. So we will also have wild type cells in the mix as well. And this is very important as, again, we want to make sure that what we are observing is from a single knockout and not from two cells with two or more targets or genes knocked out in them. Now, after we infect our cells with those lentiviruses containing our guide RNAs, and most often also containing the instructions for the cell to express a Cas nuclease, because again, remember, we need both in order to create those double-stranded breaks, we can then perform either a positive or negative selection pressure through a positive or negative selection screen. In a positive screen, the main goal here is to really identify cells that are going to survive your selection pressure. So in other words, in this case, most cells will actually die, and you will be left with a few remaining populations of cells that are going to survive that positive selection pressure, which you can then identify via Illumina sequencing by the presence of DNA detected encoding the guide RNAs conferring to the particular knockouts present. And so, for example, this can be used to help identify particular genes that may confer sensitivity to a particular treatment that, when knocked out, allow cells to grow under the specific treatment that would otherwise not be able to because their sensitivity gene or genes have been knocked out. Now, you can see that in this example, we are only detecting cells infected with the A lentivirus after sequencing, and thus that particular gene may be related to sensitivity of a particular treatment in this cell line that, when knocked out, allows the cells to survive and thus may be related to sensitivity here. On the other hand, in a negative selection screen, most cells will survive, and what we are looking for here is the absence of certain knockout cells after sequencing. And so therefore, you're going to need to sequence the starting population, apply that selection pressure, and perform another round of sequencing on the surviving cells to identify the list of guide RNAs that are underrepresented after selection. And so in other words, we are looking to identify cells that are not surviving the selection mechanism by the absence of their guide RNAs. So we can then identify cells with potential resistance genes knocked out. And you can see that in this example, after sequencing, we're not detecting knockouts B or C, but because C is most likely an essential housekeeping gene, shown in the control in this example here, gene B may be responsible for drug resistance in this particular resistant cancer cell line. Now, as we mentioned previously, you can actually purchase pre-designed libraries from places such as AdGene. And one of the most popular genome-wide CRISPR screen libraries was developed by this group at the Broad, which they call the GECKO library, or Genome-Scale CRISPR-Cas9 knockout. And so the GECKO library targets roughly 18,000 genes in the human genome, with about 3 to 4 guides per gene, or for a total of 65,000 guides. Additionally, this group actually designed their lentiviral vector to deliver Cas9, the guide RNA, and a puromycin selection marker, which actually allowed them to select for only transduced cells and to get rid of all non-transduced cells. And it's important to note that this group was successfully able to deliver both Cas9 and the guide RNA through a single vector, so they didn't have to actually first generate a cell line that expressed Cas9. They achieved this at the same time they introduced their guides. Now, in order to validate their GECKO library, this group tested the efficacy of their library by demonstrating its ability to enable both negative and positive screens. And so for their negative selection, they profiled the depletion of guide RNAs targeting essential surviving genes by transducing both a human melanoma and a stem cell line with the GECKO library. And so here, they were able to reveal, after deep sequencing, a significant reduction in the diversity of guide RNAs in the surviving cells. And so here, the absence of certain guides in their sequencing results allowed them to conclude that those were knocking out essential surviving genes. Now, in order to test their positive screen efficacy, they looked to identify gene knockouts that result in resistance to a BRAF protein kinase inhibitor, PLX, in melanoma in that same melanoma cell line. And at the end of a 14-day PLX treatment, they were able to identify the enrichment of multiple guide RNAs targeting genes that, when knocked out, could contribute to PLX resistance. And so this included both the identification of previously reported candidate genes and also a handful of genes not previously reported in PLX resistance. So they did discover some novel resistance genes with this experiment. In addition to the GECKO library, though, there are also a few other groups who have developed libraries of their own as well, including the Toronto Knockout Library and the Brunello libraries, also from the Broad. Now let's move on to discussing how we perform hit identification. Or in other words, how do we quantify the guide RNAs present after our positive or negative selection step? And so like we mentioned previously, this is going to require downstream NGS as a readout. And there are a few ways to get the pool of knockout cells ready for sequencing on an Illumina instrument. However, one of the most common methods is via amplicon PCR. Now, because that lentivirus is going to integrate its genome into the host, we are able to effectively design PCR primers basically that'll bind to that lentivirus sequence flanking our guide RNA, and so we won't need to design a unique primer for each guide itself. So we are really going to take full advantage of the viral vector backbone sequence here that's flanking those guides to make our lives much easier when it comes to primer design. Now, in this paper from the Rana lab at UCSD, they designed a set of PCR primers able to effectively amplify the constant lentiviral integration sequence flanking the guide RNAs from the GECKO V2 library. But theoretically, their method could be adapted to other libraries as well with a few minor tweaks in primer sequences. And so their approach utilizes a two-step PCR in which their first PCR utilizes primers that recognize the constant lentiviral integration sequence, followed by a second PCR to add the Illumina P5 and P7 adapters closer to the actual guide RNA itself. And so therefore, this design utilizes what's called a nested PCR to help increase sensitivity and specificity here. Now, one thing to note is that this group staggered their primer binding sites across that lentivirus sequence. So basically, they designed multiple primers with multiple different binding sites within that same sequence flanking the guide RNA. And they did this in order to help increase color balance and diversity for sequencing, because otherwise you would start each read with the same sequence, so it would have really low color diversity, and the instrument would have a tough time making base calls. And you may also want to think about Phi X here as well, just to kind of further ensure color balance. Now, once you get your second PCR product here, you're ready for sequencing. And so typically you want a read depth of anywhere from 100 to 1000x per guide RNA sequence, depending on the type of screen. And so negative screens may require more sequencing depth than positive screens because you are looking here for the absence of guides. And positive screens may not need to be sequenced as intensive or as deep as negative screens. Now, in addition to bulk approaches where we're taking our bulk pool of cells, extracting the DNA, and sequencing to count our guide RNAs, there are also single cell approaches that allow us to kind of take this one step further. And so with the single cell approach, you can identify which particular guide RNA is expressed in any one given cell, and as a result, you can also see the full transcriptional profile of that particular cell as well. And so this allows you to not only identify that perturbation, but to also look at how this particular knockout is affecting the global gene expression phenotype of that particular cell, which can then, help us better understand why our particular knockout is behaving a certain way. And so whether that be resistance or sensitivity to our experimental conditions. And one of the most popular methods of combining single-cell RNA-seq and guide RNA detection is a method by the name of Perturb-seq, which combines droplet-based single-cell RNA-seq with a strategy for barcoding CRISPR-mediated perturbations that allows many perturbations to be profiled together, making it compatible with these large screens. Now, I'm not going to go through all of the details of how this is done in this presentation, but there's companies like 10x Genomics, actually, that have some product offerings in this space that allow you to combine single-cell RNA-seq with feature barcoding technology to detect guide RNAs within the same cell. And so one thing I will note, though, here is that this workflow requires the integration of a capture sequence in your guide RNA, either at the 3' or 5' portion of that guide. But overall, I wanted to again stress the value here of this method, which is a means to analyze the transcriptional effect or phenotype in addition to that survivability of a particular knockout. It can actually potentially lead to a decrease in the cost and amount of time needed to further assay the complex effects of these perturbations in your screen. And with that, I would like to really thank you for viewing this video, and I hope that you walk away from this presentation with some of the tools necessary to begin some of these CRISPR screening experiments. And as always, Illumina is here to help, and we wish you the best of luck in beginning your CRISPR endeavors.